the best job in the world. I spend my life making pretty pictures on extremely expensive toys for all kinds of people. And it really is still true that a picture paints 1,000 words, and it always will be true. The amount of information, the amount of communication, the amount of joy you can get out from a picture is phenomenal. So I'm going to kick off for about 5 minutes and then hand over. So essentially we are an imaging facility for preclinical imaging. We don't do MRI, we don't do PET scans, that kind of stuff. But we have a range of different microscopy techniques using light, electrons and X-rays, which can cover pretty much the whole of the scale of what we want to look at in terms of biology and therefore medicine and therefore health, from the very macro end right down to the large molecule end over a scale of about 1 billion-fold, okay, in terms of the range that our microscopes can do. Different microscopes work in different ways, produce different sorts of images that tell us different sorts of things. Some are sort of anatomical, structural, some can give us functional information. So I'm just going to do a quick whiz through some of those. So, scanning electron microscopy, you often see these images. Very, very three-dimensional. They're captured in grayscale, so these are the top of airway epithelial cells and the cilia that move the mucus and all the stuff that you breathe and you don't want up and out of your lungs. A little bit of leather there. This is a petal, a high magnification, and here we've got blood cells. So very, very three-dimensional images. Often false coloured, it gives them extra wow and extra emphasis, and very large depth of field, and not too complicated sample preparation. Going much, much higher in magnification, transmission electron microscopy. This is our highest resolution image where we can see down to large molecules. But often we use it in terms of cells diagnostically and for research, giving us very, very fine detail. So those little cilia you saw in the previous one, these are cross-sections of them. So we're going down really to the minute scale. This is a nerve cell, and the spirals around it it's called a Schwann cell. It wraps around, it provides the electrical insulation for the electrical signal to pass down that nerve and not to cross-propagate. We do a lot of work on kidneys and kidney diseases as well, and this is part of the filtration unit of the kidney, where we've got some really, really interesting cell shapes going on. But all functional, all anatomical, all structural information. When we get onto light microscopy, we can put function into that. We can probe different things. We can make different things in a sample glow in different colours because they've got different components on them. So these are confocal microscope pictures. These allow us to do optical slicing in 3 dimensions, which means you can look at three-dimensional objects and go down through different layers within them and get a three-dimensional data set. Out, we can do time-lapse imaging as well. These are epithelial cells moving to heal a wound where a little scratch has been made in a sheet of cells. Everyone thinks of cells in our body as being sort of fairly inert, but they're incredibly active. And this, I was talking with Kevin over lunch about virtual microscopy, which is kind of Google Earth and microscope size. So this is a cross-section of a mouse brain. It's maybe 10, 12 millimeters across and 8, 9 millimeters deep. That's actually over 2,500 individual images done a la Google Earth. And if I kind of zoom into that little bit there, that's the kind of resolution. So this is the pixel count when this is merged is something like 87,000 by 50,000 pixels. So it's phenomenal resolution. You could paint the whole of this wall with it, you still wouldn't see the dots. And then we have three-dimensional images coming out from light sheet, which does optical sectioning. This is a bit of mouse brain again, and that one is a little shrimp where we get three-dimensional data sets out from these, which help us look at stuff, interpret stuff in lots of different ways and at different resolutions. And then we have micro CT. Not quite as high resolution, but phenomenal 3D ability to look at objects up to about the size of your fist, down to a few millimeters, and then be able to go in and analyze these. And again, this is one of the, talking with Kevin again at lunchtime, who's into AI and AI applications. This is one place where we can really do with some extra AI, because this is all done manually. It takes about 6 months to do. Data set can be generated in a day and can take us 6 months to analyze. We also do a huge amount of outreach. We go out, so next week we're going to be at the New Forest Show, of all places. Okay, it's going to probably be sweltering hot or terribly rain. We go out to sort of science events for all ages. the university's open days, hospital open days, like that. We take our microscopes out, our light microscopes out. So this is up at Winchester School of Art with their fabric design course, using our microscopes to see things in different ways and learn more things about them as well. And this is just a quote from someone who attended the university one. Microscopes are epic and amazing, and it's very, very true. They've got real potential. People love microscopes. People love looking at things down microscopes and seeing microscope images because they see things they've never seen before. We've done a lot of science-art collaborations. We're going to hand over to Anna and Leon in a second, but I'm just going to concentrate on one of the other ones we've done. These are all on our website. So Saram was an artist based in Romsey who had an Arts Council-funded project in Portsmouth, where she went around different community groups, gathering tiny little fragments from people's treasured personal possessions with their permission and with the story behind them. We took them and imaged them all on our confocal microscope. So here's just one. The website is still live. This was a jerkin from the Mary Rose. This has been under the sea for 500 years. And no staining, just putting them under the microscope and getting fluorescent images out from them. Great fun and really rewarding to do. So that's all I'm going to say, and I'm going to hand over to Anna and Leon to talk about their project they've done with us.
I shall read. Today we're going to talk about some of the work we undertook for a year-long residency that we had at Southampton University, but it was hosted by the Winchester School of Art. And this was actually during the pandemic, so the residency actually overran by, I think, about at least 6 months. Could have overrun by several years, actually, as far as we were concerned. So I'll just go through some of the details anyway. First of all, the art school at Winchester is the oldest art school in the country, and 2020, which was a pandemic year, it was its 150th anniversary, and we were invited to make work in response to this iconic building here, which is the rotunda outside the, well, it's at the front of the actual college. And we were invited to make, in response to this iconic building, and forge connections between the different faculties within the university. I can talk a little bit later about what our practice is, and it'll make some more sense of this, I think. We were really interested in exploring different imaging techniques, some of which we'd been aware of from previous residence that we did at another hospital. This was back in 2000. And the lockdowns and restrictions caused by the pandemic meant that we had really limited access to the actual art college. But by some strange series of events, we were able to get into the biological imaging unit. And so we gave our residency the title Inner Worlds, and it was our starting point was to explore the inner worlds of common materials sampled from around the campus and also around the biomedical imaging unit itself. And these use these as a basis to create some new work. I mean, in a sense, the residency that we did was the work that we created was almost like a visual diary of our processes that we actually went through during this amazing year of discovery. So we were really interested in how visual techniques developed for science and calibration, such as LiDAR scanning, which we've worked a lot with LiDAR scanning over the years, and we managed to crack into that, which was normally something used by, scientists, engineers, surveyors, and there's a whole language around it. But there's also a very big overlap for art. A lot of artists are beginning to use these techniques, and it's not the actual, we're not interested in the technology, we're interested in what the technology actually reveals. And there's a whole lot of conversations around this. This is a kind of where science and art start to sort of overlap each other and become very grey areas, very interesting areas. So anyway, we began as we often do, we work site-specifically a lot of the time. We'll generate work from whatever situation we're in and wherever we're at, and often generate it from a specific site, and then allow that to create journeys of discovery as we go along, and then we consolidate that into piece of work or an event at the end of it. So we started off, this is round the rotunda, there's a pond, and through another project we become interested in diatoms. That's another long story, there's lots of long stories. So we began collecting samples of water from the rotunda pond, and we took these over to the BIU, the biomedical imaging unit, and they helped us prepare, well actually these initial ones, they prepared these, and we were able to then examine them under some of those different techniques that were shown at the beginning. So starting off with confocal microscopy, and then scanning electron microscopy, which is actually what we ended up settling on for the majority of the year that we actually worked there. What was brilliant about scanning electron microscopy, which actually we'll get to in a bit. Yeah. So this is still the confocal.
This is like
oh yeah, right. Yeah. So what's really what we found really amazing about the scanning electron microscopy is that it's almost infinitely zoomable. It isn't really, but it sort of almost feels like that. And so you can go from this realm into, we found a way of going from this realm, which is our, the macro realm, and zoom directly into elements and areas of space that you simply can't see. But it renders them as these almost three-dimensional but monotone kind of structures. And you'll keep zooming in, and then it becomes quite sort of ordinary, and then all of a sudden you'll see something. And we spent days and days actually just exploring samples, which was phenomenal really to do.
These are the images here, aren't they?
Yeah.
And they go from 85 times magnification to actually to 1,000.
So I'll give a bit of a very quickly, yeah. So really quickly, if you go back to that first one. So here's another thing. It's a very abstract world. This. So that's about 2.4 millimeters across that whole thing there, because the times doesn't really make much sense until you can. And then if you go to the next one, so these are the actual diatoms, which are these single-cell organisms, and but by the time they're in this state, they've been kind of desiccated to a degree and then sputter-coated, which is a fine coating of palladium, I think, on this, and it's almost an alchemic process, which we're completely fascinated in the whole methodology behind it. And this is about, I would say, 0.1 of a millimetre across. So you can see how quickly you dive in. The more you dive in, it stays kind of the same, but you're still just going in and in and in. We were completely fascinated by this. So that's 0.2 of a millimetre across there. And so it's hard to actually envisage this, because our journeys were zoomable journeys. So this, we also, I don't know. Oh yeah, okay. I have to turn the page. This is why I never do this. Ad-libbing is so much easier. It's just to save time. So anyway, this is actually out the back of the biomedical imaging unit. This was where you could sneak out at the back of the hospital. And so wherever we were, that was our site. So in between scans, we would actually, two things that we would do. We would take samples from wherever, but we were also collecting. So this is just some lichen from the back of the unit. And okay, fine. And we were taking, and we were actually investigating the thresholds between the familiar and unfamiliar scales. And if you look here, if you go back a bit, so these were the sort of zooms that we took. So you imagine you can see the lichen there, but then we're not sure what this was. And as we zoomed in, we were kind of fascinated by these kind of journeys. With 2 minutes. Okay. So the other thing that we did during the
So in between the scans, they would kind of take quite a while. So we'd photograph and film around the site and undertake photogrammetry, which is kind of basically photogrammetry is like taking a number of photographs and then using software to kind of create a 3D model. So we're interested in using these different scales, and we also use the process to try and do it using the scanning electron microscope, but it didn't work too well.
It didn't work at all.
And then, so this is the actual compass itself. And then the final, well, we had a work in progress show when lockdown kind of enabled us to present the work, which was at Winchester School of Art Gallery, and which was a great space to sort of show the work that we'd developed so far, which were a series of films. And then also Dave and the biomedical team brought their amazing roadshow, which gave the students an opportunity to kind of try their own samples and use that creatively in their own work. We worked with the games design team and textiles and fine art.
People were really engaged in this, actually. And I think you mentioned it earlier, didn't it? I think as soon as people can see cause and effect, and they can see the relationship of this thing that they've got in their hand and then this extraordinary view of it, all of a sudden there's this sort of, I suppose we're all human, aren't we? And these are very human interactions with things, sort of deeply human things to do.
And then last year, actually, beginning of last year, we were actually able to present all of the culmination of that work, which was an 11-channel sound and video installation that was back projected onto the Rotunda building itself. So these are 11 windows here. We set up 11 projectors, and they were all projecting, and it was an amazing kind of support there with the technicians from the art college and also the curator Sarah Roberts, who've been our support.
So this is as we opened up after the pandemic as well. So this was all most of the time it was during the pandemic. So it's kind of an interesting time to be going on one of these journeys, really. You know, we were in this room going inwards and inwards, but then it goes out into the world afterwards. So it was a nice way to sort of move into I suppose, a sense of freedom, I suppose, afterwards.
Yeah.
I think we might finish.
And the film, if you do want to see it, it's about a sort of 11-minute film, but if you wanted to go and see it, if you go to our website and there's the Inner Art, just put Inner Worlds page, find that. You can see the film on there and lots of information about the residency, and also the Instagram page was Inner Worlds Artist Residency. So that was kind of our way of being able to engage students when we were actually, a bit locked away from them, and keep people sort of informed of the process. So